Hello. In today's class, let us learn about another important type of tissue in plants. Phloem, just like xylem, is also important. It is present in vascular bundles. Both are complex tissues made up of different types of cells. Xylem, phloem, on the whole, conduction of water, conduction of minerals, conduction of the organic solutes. Phloem is also known as leptome. We don't use the word leptome nowadays. It is used in bryophytes just like Veneria, polytrichum. Phloem is a complex tissue. It is a complex tissue made up of different types of cells. It is a conducting tissue. Conducts the organic solutes prepared by the leaves in photosynthesis, just like carbohydrates, amino acids, the simple lipids. It conducts the organic solutes in the downward direction from shoot system to root system, or sometimes in lateral direction, either downward conduction or lateral conduction. The phloem is present in all vascular plants, right from pteridophyta, gymnosperms, and also angiosperms. Phloem consists of 4 types of cells. It is a complex tissue made up of different types of cells. What are they? Sieve elements, companion cells, Phloem fibers, Phloem Parenchyma. Again, sieve elements, companion cells, Phloem fibers, Phloem Parenchyma. Sieve elements characters. What are the characters of the sieve elements? These are living cells. When do we say cells living? They must possess the protoplasm. They should possess the nucleus. Then only we say it is a living, or it should perform the metabolic functions. Living cells. Vacuolated protoplast. Large vacuoles will be present. Therefore, the cytoplasm will be pushed towards the periphery, towards the cell wall site. Young cells are nucleated, but mature functional cells are enucleated without the nucleus. Nucleus disappears, but still the cytoplasm will be present. Conduction, main function. How do they conduct? How are they able to conduct the organic solutes? Because they possess the sieve areas. So this is a sieve area. I will come to this point a little later. So sieve elements are living, vacuolated protoplast, young cells nucleated, mature cells enucleated, conduction, possess sieve areas. The sieve elements are 2 types, sieve cells, sieve tubes. With that we can understand cells, these are cells, these are tube-like structures. Sieve elements are 2 types, sieve cells, sieve tubes. Now, characters of the sieve cells are elongated, spindle-shaped, broad in the middle with the tapering ends. Then only it will be spindle-shaped, narrow ends, vacuolated protoplasm, nucleus absent in mature cells. nucleus absent in mature cells. In young cells it will be present, but later on it disappears. Then sieve areas are not well organized. Sieve areas. This is a sieve area present on the tangential wall. Then these are sieve pores through which the organic solutes pass from one cell to another cell. These are present on lateral walls or tangential walls. This is sieve area. These are sieve pores. But the randomly sieve pores are randomly distributed. Sieve areas are not well organized or not well present in pteridophytes, gymnosperms. Pteridophytes and gymnosperms. Then when we come to sieve tubes, cells elongated, cylindrical, vacuolated protoplast, nucleus absent. Cross walls, these are the cross walls. This is one sieve tube, this is another sieve tube arranged one above the other. These are the cross walls between the two cells. The cross walls contain sieve plates. See the difference here. Here we call them as sieve areas, not well organized. In case of sieve tubes, we call them as sieve plates. This is sieve plate, this is sieve plate, these are the sieve pores, well organized. They may be simple, contain only one pore or many pores compound. Simple sieve plates are found in cucurbits, whereas compound are found in Vitis. Vitis vinifera means this is a wine plant. Now, sieve tubes, cells elongated, cylindrical, vacuolated protoplast, nucleus absent, cross walls contain sieve plates, simple or. So, the difference between here they are elongated, they are spindle shaped, closed ends, sieve areas not well organized, pores are numerous, but here they are cylindrical. Cross walls contain the sieve plates. The difference, sieve plate well organized. Then companion cells. Who is companion? One who always follows a friend. Just like that. These companion cells are always associated with the sieve tubes. By the side of the sieve tubes. They are always found associated, adjacent to the sieve tubes. This point very important. Companion cells, as the name itself indicates, they are the companions of the sieve tubes, always associated with the sieve tubes. So wherever the sieve tubes are present, companion cells are present. So these two, sieve tubes and companion cells, are present in angiosperms only, sometimes found in netum or ephedra. So this is cross section. This is a sieve tube, the sieve plate cross section, sieve plate with many sieve pores. This is companion cell. It is living, nucleated, but smaller than that, adjacent to present. So remember, sieve tubes, companion cells are always associated. They are present only in angiosperm. Then phloem fibers. Fibers means sclerenchyma fibers, elongated with narrow ends, lignified, dead cells. All these characters we can apply here, but the fibers associated with phloem, present in the phloem tissue, are known as phloem fibers. Sclerenchyma cells, dead, elongated, spindle-shaped, lignified, pits will be present, provide the mechanical strength. They provide the, parenchyma cells never provide the mechanical strength. Even sieve tubes, sieve fibers also do not provide. So, in order to provide the mechanical strength, phloem fibers are present. One important point, jute, Corchorus capsularis, we get the commercial fiber. That commercial fiber is technically, scientifically, in botanical language, it is the phloem fibers of this plant, secondary phloem. Gunny bags. So gunny bags are made from jute. The jute plant is Corchorus capsularis, and that plant, after the harvest, the stems are soaked in water, allowed to ferment. Then all the tissues will be removed. That fiber is extracted commercially. Jute, Corchorus capsularis. It is technically, in botanical language, it is the phloem fiber. Phloem fiber present in the secondary phloem after secondary growth. Then Phloem Parenchyma, elongated, living parenchyma. Parenchyma cells present in the phloem. Elongated because of conducting tissue, the cells are always elongated, living, thin-walled cytoplasm. These cells store the starch, fats, tannins, resins. We can simply say sclerenchyma fibers associated with phloem or phloem fibers. Parenchyma cells associated with phloem or phloem parenchyma. Mechanical strength. Blast fibers. Here it is a living. Thus, phloem is a complex conducting tissue present in vascular bundles. Then consists of 4 types of cells: sieve elements, companion cells, phloem fibers, phloem parenchyma. Sieve elements characters. Sieve elements, sieve cells, sieve tubes. Main difference: shape. Sieve areas are present on lateral walls. The sieve tubes contain the sieve plates, well organized sieve plates with sieve pores on end walls, cross walls. Then companion cells, these are always associated with each other, present only in angiosperms. Phloem fibers, remember Bast fibers. Then these are the phloem parenchyma. Okay. Then in the next few classes, I will take about the primary structure of the root, stem, leaf, and that will be followed by the secondary growth in stem. So that is also very important. Follow me in lectures. Thank you.